Hello everyone! Today we are going to talk about transport, and today we are going to talk about transmembrane transport into the mitochondria and peroxisomes. So in our outline today, I mentioned that we are going to have 6 sections on membrane transport, and today this is our lecture on section 2, and here we discuss transport into the mitochondria and peroxisomes. So I wanted to remind you of the evolutionary scheme that we talked about last time, in which we can group the compartments of the cells into 4 families. And in section 1, we talked about the nucleus and the cytosol and how we can use nuclear pores and the nucleus that allow the transport back and forth between the nucleus and the cytosol. Today, we want to talk about mitochondria. Mitochondria, again, they're isolated from the cellular traffic. Remember, we envisioned that they were engulfed by a pre-eukaryotic cell, and that having that prokaryotic cell that became the modern day mitochondrion allowed the pre-eukaryotic cell to oxidize glucose aerobically. So oxygen became essential instead of being a poison. Although oxygen can still be damaging now, and we have ways to deal with it, which we'll talk about at the end. So for that reason, the type of transport that we'll talk about today is different. So this red arrow right here, that was what we talked about last time. That was gated transport back and forth between the cytosol and the nucleus. So that occurred in both directions, whichever was appropriate. Today, we want to talk about transmembrane transport. And notice here that the arrows are blue because it's a different kind of transport. And also notice that they're only going in one direction. So for the plastids and the mitochondria and the peroxisomes, transport occurs from the cytosol to those organelles, but it does not occur in the reverse direction. So again, right now, our focus is mitochondria. We won't talk about plastids because plants are beyond the scope of this course for the most part, but know that they are similar. So transmembrane transport, illustrated as a blue arrow and occurring in one direction from the cytosol to either the plastid or the mitochondrion, but not in the reverse direction. So here we show a mitochondrion and a chloroplast, and again I want to focus on the mitochondrion. So the way that these things are produced in the cell are a mitochondrion or a chloroplast will grow, and then fission will divide them and produce smaller organelles. So that's how they are produced. So what that means is that if they're going to grow, they need to import proteins from the cytosol. Both mitochondria and chloroplasts do have their own genomes and their own ribosomes, and they can produce some of their own proteins, but the majority of the proteins that constitute these organelles are imported from the cytosol. So that's why this transmembrane transport we're talking about today is so important. So the way that this occurs is mitochondrial proteins are first fully synthesized in the cytosol on cytosolic ribosomes. And then the transport occurs. Now these adjectives are important because it's different for different kinds of transporters. So this is post-translational. So the proteins are completely made first. So the translocation happens after they are made, post-translationally, and they are transported unfolded into the mitochondrion. So what that means is that the cell needs to have some way of distinguishing the proteins that are destined for the mitochondrion and which ones are to remain in the cytosol. So it turns out that mitochondrial precursor proteins have an N-terminal signal sequence. It's removed after the protein is imported, but that signal sequence is a little address label that tells the cell that that protein is supposed to go to the mitochondrion. So this signal sequence is typically about 18 amino acids long, and it forms an alpha helix, and it forms a special type of alpha helix called an amphipathic helix. So this is the first 18 amino acids of cytochrome C oxidase. So hopefully you remember that constitutes a portion of complex 4 in electron transport. And if you just look at the primary sequence, the artist has colored the lysine arg positively charged amino acids in red, And the polar amino acids, serine and gliadin 3, are highlighted in yellow, but when you form the alpha helix, you see that you have the positively charged amino acids on one side of the alpha helix and then the polar amino acids on the other side. So here is a receptor protein to which this signal sequence binds. So there are specific contacts that this receptor protein makes with this amphipathic helix, and an important context between the positive residues that are one side and the uncharged polar on the other side. So the translocation process involves proteins called Tim and Tom. So an amusing thing is that we have both Tim and Tom in this class. And another amusing thing is that this is not the first time this has happened. I taught this course about 15 years ago, and I also had a Tim and a Tom in my class. So this is the second time I've had both a Tim and a Tom in my class. So the Tom complex that's translocation of the outer membrane, and the Tim is translocation of the inner membrane. So the Tom complex recognizes that signal sequence that I just showed you. And the Tim complexes, there are actually a number of them, as you'll see in the different pictures I've pulled from different textbooks. Functions across the inner membrane, and in some pictures it shows the protein spanning both membranes, and other pictures it doesn't. I think maybe the jury is still out on this because I wasn't able to resolve this issue. But Tim allows the proteins to be transported into the mitochondrial matrix. There's also a complex called OXA, and that mediates insertion of the inner membrane proteins synthesized within the mitochondria. So that's the genome and the ribosomes that the mitochondria has for itself. So those are some of the players. So here's a picture. So again, Tom is translocase of the outer membrane. So we can reorient ourselves. Here's the outer mitochondrial membrane, the inner membrane space, the inner mitochondrial membrane, and then here's the matrix. So out here, we're in the cytosol. So the Tom complex consists of a translocation channel, but it also has receptors that recognize that amphipathic helix. We also have a SAM complex, so we don't have a SAM in our class, unfortunately, but SAM will be involved later on. And this is the Tim complex right here, and then this is OXA. So we want to talk about how all of these players help in the translocation process. So again, this domain right here on Tom recognizes the amphipathic alpha helix. So we can do some experiments to show that there is this signal sequence that targets these proteins to the mitochondrion. So you can do a cell-free system where you just isolate mitochondria. So here are my mitochondria, and then here's my protein up here, and the protein is shown in green with the sort of a pink signal sequence. So the signal sequence is directed to Tom, which is not shown in this picture, but it starts to thread itself through. But if you carry out this experiment at 5 degrees, that translocation process can occur. So that means that the signal sequence ends up inside, but the rest of the protein is outside. So at this point in the experiment, if you add a protease, you're going to hydrolyze the protein that's exposed on the outside of the mitochondrion, whereas the signal sequence is inside and it will be protected. So to continue the experiment, if you take these mitochondria right here and warm them up, now the translocation process can occur. The protein is imported, unfolded, and once it's inside, it gets folded. So now adding the protease means that both the protein and its signal sequence are protected. But if you do another experiment where you expose the whole system to detergent, you know from your other studies that detergent disrupts membranes. So if you disrupt the membranes, then that membrane protection is gone, and then the protease is able to hydrolyze both the signal sequence and the mitochondrial protein. So I just want to remind you that if we're transporting proteins into the mitochondria, we have 4 different places they can go. They can go to the matrix, they can go to the inner mitochondrial membrane, they can go to the inner membrane space, or the outer mitochondrial membrane. So in our discussion, we're going to see how each of these proteins can be targeted to the correct location. So let's first talk about how do we get into the mitochondrial matrix. Well, first we have our precursor protein, which again is shown in green, and there's a signal sequence that's shown in red. And here's Tom, which is shown in yellow. So Tom has the translocase, but it also has this protein with the receptor. And this recognizes that signal sequence. So the detail's not shown here, but that red signal sequence is the amphipathic helix. So the protein binds to that domain on Tom that recognizes the signal sequence, and then Tom passes it along to this translocase. So there's a pore that opens up, and the protein is threaded through, unfolded through Tom and then through Tim. And once inside, there is a signal peptidase that hydrolyzes the peptide bond right here. So now that we're inside, we don't need that signal sequence anymore, and now we have our mature mitochondrial protein. So here's another view, because this shows a few other things that we need to pay attention to. So here's our precursor protein. This artist shows the protein as blue with the targeting sequence in pink, but also emphasizes that we do have to unfold the protein. So that means we need cytosolic chaperones to bind to the hydrophobic regions of the unfolded protein so it doesn't aggregate. So cytosolic chaperones are really important to this process. Then the next thing that happens is that signal sequence will be recognized by this receptor on Tom, and then the translocase will be opened up. So open up a pore and start threading this through, and then Tom hands this off to Tim, and now the protein is threaded through both of these translocases, and the signal sequence is cleaved. But the other thing that's shown here is that now we need to fold this protein. So now we will need matrix chaperones in order to fold the protein, and we will need the hydrolysis of ATP in order to do that. So we talked about that when we talked about chaperone mechanisms earlier in the course. So there's a bind release of ATP that's necessary for this folding to occur. Luckily, we are in the mitochondrial matrix, and if you remember from electron transport, this is where ATP is generated in electron transport and oxidative phosphorylation. So we have the ATP generated right where we need it. The other thing is that this transport process itself requires the gradient, the protonic gradient that's established by electron transport. So again, it's very convenient that we have these things going on exactly where electron transport and oxidative phosphorylation are happening. Okay, so here's a picture. I have a video. I'm not going to show the video here because I did try and do this on my screencast program, and it produces a very echoey sound. So I did post it on Sakai, so you can go and look at it. But it shows the process right here, and this shows specifically these energy requiring steps. So we have the chaperones here. The signal sequence is recognized by Tom. We have the chaperones on the cytosol that have the bind and release of ATP in order to unfold the protein. Then Tom hands the protein off to Tim, and when you're doing this threading process right here, the differential ion gradient produced by electron transport is needed for this threading process. And the mitochondrial chaperones with their bind release of ATP helps pull the protein into the matrix, and makes sure that it goes in that direction and doesn't back out back into the cytosol. So all of these energy requiring processes, all of these involving the energy of the phosphoanhydrate bonds of ATP hydrolysis ensures that this process goes in just a single direction and not in the opposite direction. So that way the protein is in the cytosol and comes into the matrix, but does not go in the reverse direction. Okay, so what about if we want to go into the inner mitochondrial membrane? So I just showed you how we go into the mitochondrial matrix. How do we get to the inner mitochondrial membrane? Well, there are actually 3 ways you can get to the inner mitochondrial membrane, so let me show them to you. Let's talk about matrix targeting sequence and stop transfer. Okay, so it would be nice, I think, if there were some of these things were colored a little differently, but this shows a protein that is destined not for the matrix, but for the inner mitochondrial membrane. So if you have proteins involved in the electron transport chain, for example, they would need to be here. So you have the signal sequence that directed this protein to Tom, and then Tom starts the translocation process to Tim, and then there's the signal sequences cleaved by the signal peptidase. But this blue sequence right here is called a stop transfer sequence. So what happens is when the protein is threaded through, it starts the threading process, but as soon as it gets to that blue domain of the protein, it stops, and then it dissociates from Tim. And now you are stuck in the inner mitochondrial membrane, but that's exactly where you want to be. So I have a picture from another book. So this is, as I mentioned, this is cytochrome oxidase. So that's a protein that you would want to be in the inner mitochondrial membrane because it's involved in electron transport. So here the artist shows in pink, that's the signal peptidase that you will cleave. And then in blue, you're starting to thread the protein through the translocase, but this red sequence right here, that's a stop transfer sequence. And we shouldn't be surprised that it's hydrophobic because that's the domain of the protein that ends up being the membrane spanning domain of that inner mitochondrial membrane protein. So this diagram I think shows this a little clearer. So again, here's the protein, and this shows the translocase process, and we start to go through this through Tim. We get rid of the signal peptidase right here, and the chaperones are ready to start folding, but that red sequence right here is a stop transfer. And what we have now is that sequence right here, this hydrophobic sequence that is the stop transfer sequence, that becomes a single alpha helix membrane spanning domain of the protein. And now this is the domain of the protein that faces the matrix, and this faces the inner membrane space. But this protein resides in the inner mitochondrial membrane. Okay, how else could we get to the inner mitochondrial membrane? Well, we could use OXA1. So, here, the translocase process is shown as it was before, but here, that's why it would be nice to have different colors. This blue right here is not a stop sequence here. Maybe they should have colored it differently so we wouldn't be confused. But this is a second signal sequence. So here the entire protein is threaded through. It does get into the matrix, but you hydrolyze that signal sequence that targeted to the mitochondrion in the first place. But now you have a second signal sequence, and that is recognized by OXA. So this binds to OXA, which positions this properly in the inner mitochondrial membrane. So OXA is needed because remember, in the matrix we have ribosomes that are where the mitochondrion is making its own proteins. So those proteins need to get to the inner mitochondrial membrane too. So OXA helps with that as well. And here's an example. This is ATP synthase. So again, this is involved in oxidative phosphorylation. This would be the enzyme that couples ADP and phosphate to make ATP and oxidative phosphorylation during electron transport. And again, here, this shows that this particular protein, there's its signal sequence. This actually has two sequences recognized by OXA. So that's going to have two membrane spanning domains. So that is shown here. So if you happen to have two OXA sequences, again, this shows the transport into the mitochondrion, and then Tom handing it off to Tim, we get rid of the signal sequence, but the entire protein is translocated into the matrix. But when we have these two membrane spanning domains, OXA will recognize this, and it's OXA's job to position this protein properly into the inner mitochondrial membrane. And here's an example where we have two alpha helices and not just one spanning the membrane. All right, we have a third one. What if we have not just one or two, but multi-pass membrane proteins? How does that happen? both of the uh, uh, diagrams are, however, a little cartoony, which tells me that maybe we don't know exactly how this happens. So I'm going to call this path C. So I referred to paths A and B before. So what happens is the translocase occurs, and what happens is there are intermembrane space chaperones that hand this protein off to Tim, and then this is responsible for positioning the protein into the intermembrane protein into the intermembrane properly. So this sequence that's shown in red here is an internal um, signal sequence. It's not at the end terminus, it's internal, but it does eventually get to Tom, even though it's internal, but it helps Tim recognize how we should fold this. So I have an example here. So this is the ADP-ATP antiporter. So you might remember this again from electron transport. If we're generating all of the ATP, or not all of it, but most of the ATP of the cell in the mitochondrion, we are going to have to export some of it into the cytosol where it is needed, and we'll have to import ADP as a raw material to make the ATP. So that happens in the ADP-ATP transporter and antiporter, and it is a multi-pass protein. So again, different colors might be nice because that's that internal sequence that's recognized by Tom, but then Tim has to recognize them too to make sure that this is placed in the membrane properly. So this shows this, maybe a little bit clearer, maybe not, but these shows the internal sequences recognized by the receptor on Tom. Tom translocates this, hands this off to Tim, and then this is what the final assembled protein looks like. So all of these internal targeting sequences become the alpha helical transmembrane domains for this protein. Okay, so we got into the matrix, we got into the inner mitochondrial membrane. How do we get to the inner membrane space? Well, what we can do is we can borrow those pathways I just showed you, A, B, and C. So if we get to, if we use A to get to the inner membrane space like I showed you here, or with B where we involved OXA, what we can do then is once these proteins are anchored in the inner mitochondrial membrane, you could have a protease that cleaves it, and now this protein is free in the inner membrane space. So here, to target a protein into the inner membrane space, basically we're borrowing pathways A and B to get the protein into the inner mitochondrial membrane, and then yeah, that should say inner mitochondrial membrane right there. I should fix that. Whereas then the signal peptidase then cleaves the protein, and that releases it as a soluble protein in the inner membrane space. Because remember, this this domain right here was a hydrophobic membrane spanning domain. So here's another look at it right here, where we have the targeting sequence right here, and that's the inner membrane space targeting sequence. So this protein, again, we go through the translocase, we cleave that sequence right here. We do have the protein that's initially anchored in the inner mitochondrial membrane, but then right here there's a recognition sequence for a protease that cleaves it, and now we have a protein that is in the intermembrane space. So here's another view of how that could happen, where you don't even use the inner mitochondrial membrane at all, that you just transport your protein and then it just goes into the into the inner membrane space and stays there. You would need, for this particular example, proteins that help ensure that the disulfide bonding here is appropriate. And for this particular example, these are TIM9 and TIM10. These proteins actually will escort other proteins to inner mitochondrial membrane translocators to make sure that they get to the proper place. Okay, so we got into the matrix, we got into the inner mitochondrial membrane, we got into the inner membrane space. How do we get into the outer mitochondrial membrane? Well, here we are going to use Sam. Okay, so we'll involve Tom, and again, Tom will recognize the signal sequence and start the translocase, and then the protein will be greeted by chaperones that will assist in the folding, and then the SAM complex. SAM complex job is to ensure that the fully folded protein ends up in the outer mitochondrial membrane. So there's some kind of sorting process right here. So SAM is sorting and assembly. That's what that stands for. The details of this mechanism aren't known, but these are the players, and this is as much as we know about the process at this point. The membrane transport proteins I showed you previously were alpha helices. This is how a beta porin will be positioned. And remember, the porins are in the outer mitochondrial membrane. Okay, so today's journey again was how do we use transmembrane transport, which is shown in blue, to get proteins to the mitochondria, and we won't talk about plastids, but the process would be similar. So the next thing we want to talk about is peroxisomes. Again, we'll use this transmembrane transport from the cytosol to the peroxisomes in one direction, not the other direction. So peroxisomes again grow by fission. So you'll have these vesicles will bud off from the ER and then import specific proteins that gives these vesicles their identity as peroxisomes. So here they're not like mitochondria or chloroplasts. They have to import everything. So right here you have just a little sort of membranous vesicle. So what makes this thing a peroxisome is the ability to import the proper proteins into this organelle that makes it function like a peroxisome. And then when it gets big, then you just use fission, and then you have more peroxisomes. So eukaryotic cells have these organelles, and they have high concentrations of oxidative enzymes. So to put peroxisomes in an evolutionary perspective, they're a vestige of an ancient organelle to serve to decrease the concentration of oxygen. So that would have been toxic in the early evolution. But then once the cells evolved and they acquired mitochondria, what happened is the mitochondria took over the task of oxygen utilization, and cleverly you was able to figure out how to use oxygen to generate energy, not just to detoxify it. That function of peroxisomes isn't required anymore. So basically the modern day peroxisomes carry out tasks not taken over by our modern day mitochondria. So peroxisomes contain enzymes that use oxygen because oxygen is vital, but it is damaging. The product is hydrogen peroxide, which is also damaging, so that needs to be converted to water. So you convert it to something that's less damaging. Peroxisomes also carry out beta oxidation. So you might remember both from Biochem 1 and those of you who are with me in nutrition. So beta oxidation is how we oxidize fatty acids. And we know that we do this in the mitochondrial matrix in mammalian cells. But if you are a yeast or a plant, the peroxisomes is where beta oxidation takes place. Peroxisomes also catalyze the first few reactions of plasmalogen synthesis. So I have an example to show you there. So the peroxisomes are required for the first part of fatty acid oxidations if the fatty acids are very long. So if you have fatty acids that are greater than 22 carbons, then what happens is the peroxisome shortens them, but in so doing uses oxygen as an acceptor for protons to convert this single bond into a double bond. And then catalase, which is an enzyme that resides in the peroxisome, is able to convert this to oxygen and water. So once you've shortened your very long chain fatty acids, then they can go to the mitochondria, and then the mitochondria will take over and go through complete beta oxidation. And this is the plasmalogen I was telling you about. So plasmalogens are interesting. This looks like a phospholipid, and it is. We've got the two tails and the phospholipid head group, but here you've got an alpha-beta unsaturated ether linkage. So the plasmalogens, their function isn't well understood, but they're abundant in myelin sheaths that insulate the axons of nerve cells. So I know that there's an interest in neurobiology in this class, so I thought you would be interested in that. So plasmalogens are made in peroxisomes. So I am going to bring up plants just for a minute here because the peroxisomes are involved in carbon dioxide fixation in leaves, but in germinating seeds, remember we talked about the glyoxylate cycle in Biochem 1, and we said that the glyoxylate cycle took place in the glyoxysome. So the glyoxysome is actually a specialized peroxisome. So again, this happens in seeds, germinated seeds in plants. And again, mammals don't have these. We have peroxisomes, but we don't have glyoxysomes, and we are not able to take fats and convert them to sugars. That's something that we can't do. So how do you get to the peroxisome? Okay, so if you are one of those enzymes that is required to detoxify things like hydrogen peroxide. So here the signal sequence has been worked out. So there's a peroxisomal targeting sequence, and it has an SKL sequence, and it's on the carboxyl terminal, not on the amino terminal. So this is SKL, this is serine, lysine, leucine. So it's on the carboxy terminal, and then that binds to a cytosolic receptor called the PEX5 receptor. And then that directs these proteins here to these proteins right here called PEX14. And then PEX5 returns to the cytosol to be reused. And then the PEX14 helps transport these peroxisomal proteins into the matrix of the peroxisome. And again, the PEX5 is returned for recycling. So this is a little bit different. The peroxisomal proteins are transported completely folded, whereas the mitochondrial membrane or matrix proteins were transported unfolded. So that is our journey through section 2, how we transport proteins to the mitochondria and the peroxisomes.